Alright, ladies, this is the first portion of our cellular and molecular biology notes. This first part 1, is going to be 3 different videos with one PowerPoint that you'll have, and this is totally going to be independent study. Most of this stuff should be review from your freshman year biology class, so we're going to spend our class time doing labs and dealing with some clinical medicine stuff instead of actually sitting through lectures. So it will become your homework responsibility to cover these notes on your own. Now, cellular and molecular biology is a huge, huge field. I mean, anything that has to do with cells or smaller could be lumped into this. So that means that really even other things we'll do this year, like genetics or things like that, could also fall under this umbrella if we wanted it to, but we're just going to use this cellular and molecular unit to really talk about the cells. And that's going to be our major focus. So we have 3 parts to cellular and molecular biology. Each of those will have a somewhat small test. So basically I've broken this unit down into 3 parts so that you have to kind of learn smaller chunks of information rather than just a huge test over one big thing. So this video is going to just look at the first portion of part 1. So we'll be in part 1 for the next 2 weeks here. Alright, so our essential questions for this first part of cell Molec is, how does a cell maintain homeostasis, which really will kind of bleed over more into part 2 of cell Molec. So the major focus then is this top one, which is how are cell structures adapted to their functions. And we'll talk about this more and more here as we move along, but kind of like we saw in Unit 2 with enzymes, and we said they have to have a certain shape. Everything in a living organism has a particular structure, and that structure that structure is very, very important because it allows for the function of that cell or that organism or whatever it may be. Vice versa is also true then. You will not have a function without the correct structure underlying it. And so that's a huge kind of big idea in biology as a whole, and it'll be super important here as we talk about cells. Okay, so first thing we're going to do is take a look at just a little bit of the history of the cell theory. Now, you do not need to know dates, okay, that's always true in my class, but these are just here kind of to give us reference. If we go all the way back to the 1600s, you can find the first time that someone found a cell and used that term. And the person responsible for this is Robert Hooke. Robert Hooke built his own microscope, like you see in this little picture on the left, and what he looked at was tiny slices of corks. That's kind of a fun story. We won't go into great depth, but Hooke was a teacher at a monastery, and most monasteries have some sort of business that runs with them. For instance, St. Mildred Arch Abbey up in Indiana has several different businesses that they run, and essentially they run those businesses to help pay for the monastery. And so one of the big ones in Indiana is Abbey Press, you might have heard before, that publishes books and things, or Abbey Caskets, which is a little more morbid, but they build caskets, urns, that kind of thing. But Robert Hooke worked at a monastery where the major kind of job was winemaking. And so he had access to tons of corks used to stop wine bottles, and so that's what he got a hold of to do his experiments. And what he knew was cork is tree bark, basically. So he sliced tiny thin sections, looked at those pieces of cork, and noticed these little boxy looking structures. Now, also at a monastery, the students that went to school there lived in dormitories, as we would call them today, but instead of calling them dorm rooms, they called their rooms cells. And so when Hooke looked at this under a microscope, he said, wow, this looks like the cells that we live in. And there you have it. The name stuck. Now, also in a similar time, you had a guy named Leeuwenhoek, or Anton van Leeuwenhoek. He's pictured here on the right. And Leeuwenhoek was the first person to, again, kind of build his own microscope and observe microorganisms moving around. So he was looking at pond water and those sort of things, and noticed little tiny swimmies that moved around. Now then you fast forward quite a bit to a guy named Theodore Schwann, who I think looks like Teddy Roosevelt. And Theodore Schwann discovered the cellular makeup of animals. So he's the one that told us that animal tissues are all made up of different cells. Now, why did this take 200 more years to arrive at? Because as we will see in our lab, animal cells are hard to pick out. You know, plant cells have a nice wall in between them, that kind of thing. Animal cells just kind of all blend together. So it took a long time for scientists to be pretty certain that animal tissues are made of cells. Now, the reason I have this picture here too is that Schwann is actually a little more famous for discovering something called a Schwann cell a few years later, which helps your nerves function correctly in your nervous system. All right, so that leads us to the cell theory, which there are very different interpretations of the cell theory. Sometimes they say there's 3 parts to it, sometimes just 1 part. Our textbook talks about a 2-part theory. The first part being that cells, or products made by cells, are the units of structure and function in organisms. So in other words, kind of the Mr. Burton definition, all living things are made up of cells. Second piece, all cells come from pre-existing cells. So basically, for you to get a new cell, you're going to need a cell for it to come from. Now, if you can think back to freshman year, we talked about spontaneous generation and how Francesco Redi and Louis Pasteur, which is this guy, helped to eliminate the idea that living things could come from non-living things. Right? So that does not happen. We do not get living things from non-living things. We get living things from pre-existing living things. This is just a cool picture here. This is a cell we'll learn about later this year called a macrophage. Big pink part or purple part here, and it's actually engulfing bacteria right here. So these little yellow guys are bacteria, and the big purple cell can swallow them up. Now, cells come in a lot of different sizes. The average cell is about 10 to 20 micrometers, so way tinier than our rulers can measure. Usually when we talk in terms of microscopes, we call them microns. So we'd say that an average cell is about 10 to 20 microns in diameter. But there's a huge range, right? An ostrich egg, about the size of a regulation NFL football, is a single cell. Neurons in the body, right, can run from your hips down to your big toe. So in some people, about a meter long, right, almost 3 feet long. A single cell can traverse almost the entire height of a tree. So if we're talking about a giant sequoia, we're talking about 300 feet or 100 meters. So cells can be huge. Cells can also be teensy tiny, right? Some bacteria are only about 1 micron in diameter. You can see in this range. So you would need a microscope to see those, of course. But the average cell, somewhere between 10 and 20 microns. Now, cells come in many different types, and that's going to be kind of idea number 1 for our cell Molec, is that you got a couple different types of cells out there. Now, cells come in all different shapes and sizes, many different forms, and there is a function intricately tied to that structure, right, that shape. So a couple of examples. First, we mentioned xylem and phloem last unit as the thin tubes inside of a plant that allow fluids to move up by capillary action. Here's a cross section, so it's like we cut the tree off, of xylem and phloem. Each of those is actually a cell, a long tube-like cell. Think of like a drinking straw. And so that's a cell, and its job is to move fluids long distances. So it's long and skinny like a straw. Now here's some cool cells right here. This would be in your kidney, and this is an electron micrograph, so we're using a much stronger microscope than our light microscopes that we have in class. And these cells are sort of wrapping around a blood vessel, a tiny blood vessel, in your kidneys. This is called a glomerulus. You don't need to know that term. But these little finger-like projections are all coming off of this cell, which is just wrapping around. I was thinking about wrapping your hand around something. This serves to help filter your bloodstream. And so we form sort of like this sieve here. Like if you interlace your fingers together, that's basically what we've done here. So as the fluids enter or leave that blood vessel, they can be filtered out by the kidney. That's a really specific shape, right? A really specific structure to do its job, to do that cell's function. Now, we have two large groups that should sound familiar to you. Two basic types of cells, the prokaryotes and the eukaryotes. So that's where we'll start. All right, prokaryotes. Prokarya means before the nucleus. So prokaryotes are usually unicellular organisms, so single-celled, but they grow in colonies, like you see in this picture with these bacteria. Colonies could be clusters of bacteria of these prokaryotes, chains. They form a film. But generally, the prokaryotes are thought to be the bacteria, right? Bacteria, the simplest living cells we're aware of. For our class, even though this isn't really accurate, we can kind of interchangeably say prokaryote or bacteria. Okay, they're pretty much the same deal. That's not quite true when you get to the college level, but it's good enough for us. Now, the important things about prokaryotes is what they are missing. Right? Prokaryotes, again, it means before the nucleus. So prokaryotes lack a nucleus. Right? They don't have a nucleus or a membrane-enclosed structure where we can find their DNA, their genetic material. In fact, they pretty much lack all of the membrane-enclosed organelles. So all the organelles that we would find inside your cells that we'll learn about later on this week, they're not going to be found in these prokaryotes. The one structure they do have is ribosomes, but we'll come back to that more later on. Now, a few more structures that you might see in a prokaryote. Every prokaryote actually has a cell wall, which you might recall is more so usually kind of thought of as being a plant thing, but prokaryotes or bacteria have rigid cell walls. Those are made up of carbohydrates, proteins, and lipids, but they're not made up of a certain carb called cellulose, which we'll come back to later, but cellulose is seen in plant cell walls, but it's not seen in these prokaryotes. Now, they also still have genetic material, right? They have DNA. They have a single circular chromosome of double-stranded DNA. So their DNA kind of looks like this. It is double-stranded. It is a twisted helix, but it's just a ring of double-stranded DNA. Now, you're going to find that DNA not in a nucleus, because these guys don't have a nucleus, but in a nucleoid region. So some area, wherever we find the DNA in the bacterial cell, we call that the nucleoid. And then bacteria will also have something that we're going to learn more about later this year called plasmids, which are extra little pieces of DNA. And that's actually critical for bacterial function. But again, we'll come back to that later this year and not worry about that too much right now. Now, the last thing you want to know about prokaryotes is that they can have appendages. They can have flagella, which is a whip-like tail. And a flagellum on a bacterium is like a tail that sticks out and it spins like a propeller. So it's a long tail, but it actually spins at its base rather than sort of like flapping or something like something that swims. The other thing they can have is a pilus, or the multiple there would be pili. These are sexual appendages. Bacteria are able to transfer DNA plasmids from one bacterium to the next. So if this one has a plasmid, it can actually hand it over to another bacterium. And the way they would do that is via a pilus. So they basically make this little like bridge between the two, and that's what a pilus is. There's a basic picture. This, as an example, would be the bacterium E. coli. And so if you look at this, you see the genetic material in here. And so that's labeled as the nucleoid region because that's where the DNA is. They've got a cell wall here. Oops, I didn't mean to cross it out. They got a cell wall. They still have a cell membrane. They will have ribosomes, like we said. These little just kind of short arms sticking off of it are the pili. And then notice it has this big whip-like tail as well, which is the flagellum. Now if we switch to eukaryotes, eukarya means true nucleus. So these cells, and I'm jumping to the bottom of the slide here, but these are cells that do contain a nucleus, or a membrane enclosed structure where you can always find the DNA of the cell. And in a eukaryote, that DNA is going to be in linear chromosomes, right? Linear chromosomes, double-stranded DNA. And we'll come back to that a bunch more later this year. Now, generally, eukaryotes are going to be in multicellular organisms. So they can be single-celled, something like an amoeba, but generally they're going to be multicellular organisms. They all have a cell membrane. In fact, every cell, eukaryote and prokaryote, has a cell membrane. But cell walls only show up in some eukaryotes, and that would be the plants and the fungi. Now, the other really important thing, so these two things are really important, about eukaryotes that you might remember is they have lots of different membrane-bound functional parts known as organelles. So organelles, internal compartments of a eukaryotic cell. Now hopefully you remember that we are eukaryotic, so all of our cells would have these types of structures. Now this is the slide to study if you're studying later, because what I've done is I've just made you a table of all the things that I've been talking about. So this combines all that information so that you can very quickly and easily compare prokaryotes to eukaryotes. And so to kind of run it down again really fast, prokaryotes are the bacteria. They have a single circular chromosome found in the nucleoid region. They do not have membrane-bound organelles. We didn't say this before, but they don't really do mitosis, but they do divide. They do not have a cytoskeleton, more on that later. They do have cell walls, but never with cellulose. They have small ribosomes, and they something else we'll talk about later. They have proteins in their DNA, but their DNA is not wrapped around anything special. Eukaryotes, which are our cells, right? These could be animals, plants, fungi, and the protist, which would be single-celled organisms. We've got multiple linear chromosomes, which are usually paired up inside the nucleus. We do have membrane-bound organelles. We do mitosis for cell division. We do have a complex cytoskeleton, again, more on that later. Sometimes have cell walls, which contain cellulose. We've got bigger ribosomes, and we have something our DNA is wrapped around called a histone protein, and we'll come back to that later as well. So here's a basic animal cell. This comes from the textbook that Dr. Wong and I put together. A lot of times these cell pictures are like try to be so fancy in textbooks and online and three-dimensional and all this kind of stuff, and so we decided, we just want a really basic 2D picture. So that's what this is, and it's got all the different organelles that we are going to learn here in the coming days. Here's our version of the plant cell. Notice it's boxier because it has a cell wall around it. Also notice it has this central vacuole thing, which takes up most of the space of the cell. It usually holds water. Also notice we got chloroplasts in here, which are your areas for photosynthesis. Otherwise, structurally very similar to the animal cell. Okay, here's a picture of the compound light microscope. I posted that handout on Edmodo for you to see as well, but just make sure you know what these different parts are, what we use these for, and things. I'm really just interested in making sure that you can use a microscope, but here, this way you've got this. And then this is probably more important about the microscope, and we'll talk about this as we do our lab. But realize when we talk about total magnification, this is a compound microscope. So we've got two things that we're actually putting together. And in this case, we're talking about two different lenses. All right, we've got an eyepiece lens, which is always 10x, and we've got optional or objective lenses to choose from. Not optional, you're always going to have one of them, but objective lenses to choose from on the rotating turret. And for our microscopes, although these can vary on other microscopes, we're going to see a 4x lens, a 10x lens, and a 40x lens. This would be low power, this would be high power, and the medium in between. So if we're asked for the overall magnification or the total magnification, you have to multiply those two lenses together, this one and whichever one of these second ones you're using. So if you are at low power, you would do 10 times 4 would be 40 total. If you were at medium power, you do 10 times 10, or 100 total. And if you were at high power, I know my handwriting's terrible, you would do 10 times 40, or 400, as it says here. So just keep that in mind. If you're asked about total magnification or overall magnification, always multiply your objective lens times your eyepiece lens. Okay, that takes care of the first section of Cell Molec Part 1.